Now let's look at the protein structure prediction, or how to predict protein structures. So we have three general ways to predict protein structures. So the first one is comparative modeling. So in here you have your comparative modeling is currently the most popular approach. Especially if you have your target sequence with a known homologous protein, or your protein that you want to predict, the structure of the protein you want to predict, that protein has a homologous relative that have a known structure. So in this case, you have a template. So your target, you look for a template that is usually it's a related in its homologously, or a related protein from a different organism, and you align the sequence of your target with your template, and you build a model through that. Now that is comparative modeling. Now the next one is threading. Threading is where you do not have a good enough template. There is no closely related protein that you can use fully as a template, but you can see that your proteins have segments, and those segments of your protein sequence, you can match them to a library of the known folds of the protein. So you thread your target protein based on the different sections of your or segments of the sequence. You look for the known folds that basically it's like a local alignment search tools. So like you have your you thread them to the known folds, and then you assemble those folds to form your model. So that's your fold recognition, or rather your threading segment. Now the last one is ab initio. This is the case where you have absolutely no idea. You have absolutely no template you can use. So you are going to predict the protein from scratch. So in this case, you are going to actually do Lewinthal's paradox. You're following the Lewinthal's paradox. Rather, you are trying to predict your protein through its energetic free energy minimum. So the protein's native structure is known to be the minimum energy conformation of your polypeptide sequence, and you're going to search for that. So you search it, you can look for tertiary structures. That actually the best way is actually the Rosetta method. We will be discussing that later on. Now, let's discuss first in detail the comparative modeling. Comparative modeling, you have a template. Usually, this template is a closely related protein with a known structure. So first, you need to select a proper template. So you search for a homologous protein sequence or structure, and you use different tools such as your BLAST or the Delta BLAST. Or you can also query your target against the PDB database. You have the CAT and the SCOP database. So these are the databases of protein structures. You can query that, a homologous protein, using the sequence of your structures from your to get your template. Then structurally conserved regions and structurally variable regions are identified. So it's common for structurally variable regions to correspond to loops and turns, often at the exterior of the protein. So this is usually done when you have a lower sequence similarities. So usually you target the areas where those loops and turns are at the general exterior of your protein. So basically, the exterior of the protein, when it falls into its tertiary structure or even a quaternary structure, the exposed regions are those that are mostly variable. And then once you have chosen your template, you align it with the template with your target, and you have basically you need to know the sequence similarity, the primary structures sequence similarity. For a 30% sequence identity between your target and template, the two proteins are likely to have a similar structure if the length of the aligned region is sufficient. So that means you need to have a more than 60 amino acid that is well aligned with each other. So it's good to look for multiple sequence alignment. For example, this segment matches well with this template A, and then another template matches well with the remaining another segment of that. So you can use multiple sequence alignment to actually deduce proteins from actually multiple templates. So you build your model from your template. So the variety of approaches are employed, such as rigid body assembly or segment matching, in the case where only certain segments of your template closely matches with your target, and then you are using a different template for another segment of your target. So you have segment matching, and then finally assembling it. And then finally you evaluate your model. So we have several we have several principal types of error that occur in comparative modeling. This is identified in a paper by Marty and Renom in 2000. So first is the errors in side chain packing. So remember, the side chains, your R groups. So depending on your R group, they might have clashes, because especially if you have lower sequence similarity, so there might be some steric clashes with the side chains. And then distortions within correctly aligned regions. Again, sometimes the dihedral angles, the angles and the torsions are not does not align well. Errors in regions of a target that lack a match to a template, especially notable in for example, if your target is a mutated protein and you are aligning it with the native, there might be some errors in the region where of the mutation itself. Errors in sequence alignment and the use of incorrect templates. The most common errors, of course, incorrect templates. So you should take extra care in choosing your template. Model evaluation. So once you have built your model, you need to evaluate your model. So you need to assess the quality through the band lengths and angles. Are they appropriate? So it's better to use Ramachandran plot now to evaluate, are the folds are the psi and the phi angles are in the allowable regions. The peptide bonds are planar. So do you form planar peptide bonds? Because it's an absolute rule because of the carbonyl the sp2 carbons. The carbon backbone conformations are allowable. So again, using your Ramachandran plot. And then there are appropriate local environments for hydrophobic and hydrophilic residues. For example, you are the hydrophobic residues in the interior side and the hydrophilic on the outside. Are there any hydrophobic residues exposed to your solvent? And again, of course, solvent accessibility. So again, it has something to do with the hydrophobic and hydrophilic issues of your protein. And another thing is is your protein transmembrane protein? Because depending on its position in a physiological position, for example, for a transmembrane protein, you have a segment on the outside here that should be hydrophobic, the ones that are interacting with your lipid bilayer. So you need to consider that. Now, these are the different websites for structure prediction by comparative modeling and its quality assessment. So the most common, or one of the more popular one here, is actually Modeler from Andres Salis. Actually, that is the one that we will be using, or rather we will use that in our laboratory activity. You can also do Swiss Model. It's actually an easier alternative. This is a server-based alternative to Modeler. Okay, so the rest is ProCheck, Verify 3D, and What If. These are more of quality assessments tools. And then the other option for protein structure prediction is fold recognition, or the threading. So this is useful when your target sequence of interest lacks an identifiable sequence match and yet may have folds in common with proteins of known structure. So your target might assume a fold that occurs in a characterized protein because of convergent evolution, or because the two proteins are homologous but extremely distantly related. So basically, in here, you do not have a really good enough template to use. So the input sequence in threading is parsed into subfragments and threaded into a library of known folds. So the scoring function allows an assessment of how compatible the sequence is with known structures. So basically here is this table allows you to choose what type of technique you're going to use based on sequence identity or sequence similarity of a known structure. Or rather, if you have a template, you want to use, and you compare the sequence similarity. So based on the sequence identity, what type of model would I use? So for example, for a 100% sequence identity, you basically, of course, have a good accuracy and resolution in your model. So this is the technique is done. Of course, the most accurate structure determination is X-ray crystallography and NMR. Now, if you have a less stellar or rather lower sequence identity, so your accuracy is less. So you have a 95% accuracy. You have your comparative protein structural modeling, and then lower accuracy, lower sequence identity. That lowers your accuracy. You have threading, and the lowest accuracy is the Denovo structure prediction, that is ab initio. That is basically you have no template whatsoever to use as a guide to in structure determination. Now this is ab initio method. This is your template free method, the Denovo method. So this is based on two assumptions. All the information about the structure of the protein is contained in its amino acid sequence, and a globular protein falls into the structure with the lowest free energy. So you are going to look for all of the possible combination or structural possibilities and choose the one with the lowest free energy. So in here you have your this is the Rosetta method for ab initio protein structure prediction. So from your query sequence, you fragment it. Actually, it's actually related to threading. You fragment it and look at possible assemblies. Do you form beta sheets? Do you form what are the suprasecondary structures that you might form? And this fragment assemblies is computed which is the most probable using Monte Carlo assessment. So you use coarse grain energy function to actually assess the free energy of each domain or folds, and the low resolution models, you form low resolution models from coarse grained energy function. So basically, from your fragment library, you assemble the fragments into all of the possible structures, and you calculate the energy of those. You choose the lowest energy from coarse grained calculation. When you say coarse grain calculation, this requires lower computational resources, but again, the models here are of lower resolution. So you want to actually, it's like filtering which is the best structural model from all of the possible models you have. Now, coarse grain is, of course, you have a filter is rather coarse, not fine. And then after getting for example, top 10, top 20, top 50 from the coarse grain energy function, you get an all atom energy function. So you basically need to calculate the energy using all of the atoms involved in your sequence, and you have a higher resolution models. And the lowest energy from that is the most possible model or structure of your protein, because we are assuming this is based on assumption number 1, that is rather assumption number 2, it must have the lowest free energy. So in here, in ab initio protein models, this involves molecular dynamic simulation to get the energy, the free energy calculations for each of your protein sequences. And ab initio is the one with the most computationally expensive of all of the structural prediction methods. So why do we need to predict the structure? One of the applications of your structure prediction is on drug design. So if you have a structure, you can do structure-based docking, and you can infer a possible drug candidate for a certain receptor or your enzyme or a target protein. So that's it for for this laboratory part of the lecture. So this is the reference is Jonathan Prevsner's Bioinformatics and Functional Genomics 3rd edition. So please see the laboratory activity uploaded in your Blackboard.